Hi everybody. Now today we came across the genome organization in bacteria. Genome organization, when we talk about that in bacteria, we expect that bacteria has a single circular chromosome and it is compactly arranged within the small size of the bacteria. That's what do we expect. And but we cannot generalize things like that because there are a wide range of bacteria and some of them show different variations also in that. So today we are going to I'm going to talk about small variations in the genomes of certain bacteria. Of course that would be quite interesting also for your information. So let's see what's that. So first of all we have is agrobacterium. Okay, there are lots of examples. I've just cited a few of them for your reference. So agrobacterium, in the case of agrobacterium, it has been found that it is having a it has two chromosomes basically. One is a circular chromosome and the other one is a linear chromosome. Okay, so basically the number of having one chromosome in prokaryotes is being broken with the help of it has been changed in the case of agrobacterium. Then if you're going to consider Vibrio cholera. Now Vibrio cholera, it is a causative agent of the disease called cholera. Here it is having two circular chromosomes. Okay, same way in agrobacterium, as we told, it is having two chromosomes here, but both of them are circular. And one of these chromosomes of Vibrio cholera, it contains the genes involved in metabolism and virulence, and other genes, the other circular chromosome will contain the essential genes. So without both of these chromosomes, it cannot survive because its metabolism and virulence is coded by one circular chromosome and all the essential genes are in the other chromosome. So it becomes essential for Vibrio cholera to have both the chromosomes together. But here also a violation that prokaryote has a single chromosome is violated here. Then in the case of Bacillus subtilis, okay, you can find that here it is similar to that of E. coli. It is having a single circular chromosome, that is all true, and it is occurring in the form of nucleoids as in the case of E. coli, but it uses a different architectural protein to form so in Bacillus subtilis. So this is a reading which I got from Nature Education and I've quoted it for your reference. If you want to read more, you can just go through that. A still more interesting example comes from Borrelia burgdorferi. So Borrelia burgdorferi is a bacteria which lives on the deer ticks. Okay, and it is transmitted. So when the deer ticks gets transmitted from one deer to the other, this bacteria also gets transferred from there. Okay, and it has been studied that it contains about 11 copies of a single linear chromosome. It has only one chromosome, okay, but 11 copies of that linear chromosome has been present in the case of Borrelia. So this is against, we have a concept that bacteria has only, it is found to be haploid in nature, that is, it has only one set of genes, it has only one single chromosome. That's what we consider in the case of bacteria. But in the case of Borrelia, you can find that they have a single linear chromosome, but 11 copies of that linear chromosome has been present. And another feature of this Borrelia is that unlike other E. coli and E. coli, we find that we always have a concept that DNA is being supercoiled and so that it becomes into a tight ball within the nucleoid. Okay, but the linear chromosome of Borrelia cannot be supercoiled into a tight ball. Okay, and here the strands are being found diffused throughout the cell. I can just show you the paper which showing the picture, because I cannot cite it because of copyrights, but we can just go browse and see how that picture looks. See, this is a paper from the Annual Review of Genetics, and this is a picture of Borrelia linear genome, which they have been quoted in this particular paper, and you can see that it's just diffused out. It's in 2009 that they have launched this paper, and it's just when the DNA was being, when the chromosome, see, the germ agglutinin, okay, it was being stained with wheat germ agglutinin, LX flora. What happens? A live Borrelia burgdorferi was being stained like this, and they got an image. This is being quoted from Brizan et al. I just browsed it. See, this is a paper which you can get online, and it's on the annual reviews of the genetics of Borrelia burgdorferi. You can just go through it. It would be helpful if you want to, if you are curious about it. And one more variation that we can come across is in the case of Archaea. Okay, in Archaea bacteria, it has been found that the DNA molecules of Archaea, they are composed of single cell non-bacterial prokaryotes, and they share many similarity with the eukaryotes, and they can be negatively supercoiled as well as positively supercoiled, and even not supercoiled at all. And it has been important that they are the only ones which have proteins like the eukaryote, like histones. Okay, that is, in the DNA organization, eukaryote like histones have been present in this, and rather than architectural proteins described above to condense the DNA molecules. And this acquisition of histones by archaeans is thought to have paved the way of evolution for larger and more complex eukaryotic cells. So a little more features of nucleosome and all that which have been present in Archaea, we will be explaining that when you study about what in the case of eukaryotes. In eukaryotes, you will be learning about the histones, and so that eukaryote like histones are also being found in Archaea. So what is a comparison of the prokaryotic genome and the eukaryotic genome? Now, in the case of prokaryotes, generally speaking, we contain a single circular chromosome, and eukaryotes, usually they contain a multiple linear chromosomes. And prokaryotes, they are the chromosome is condensed into the nucleoid via DNA supercoil and the binding of various architectural proteins. And in eukaryotic chromosomes, they are condensed in a membrane-bound nucleus via the histones. Because prokaryotic DNA can interact with the cytoplasm, the transcription and translation normally occurs simultaneously in the case of prokaryotes. But in the case of eukaryotes, transcription occurs in the nucleus and translation occurs in the cytoplasm. And most prokaryotes contain only one copy of each gene, that is, they are haploid. But most eukaryotes contain two copies of a gene, that is, eukaryotes are considered to be diploid when they contain two copies of a gene. Of course, there are variations. It's not that all eukaryotes are diploid, but they contain usually different copies of a gene. Non-essential prokaryotic genes are commonly encoded on the extrachromosomal plasmids, but extrachromosomal plasmids are not commonly present in the case of eukaryotes. And prokaryotic genomes, they are efficient and compact, containing a little repetitive DNA. But in the case of eukaryotes, they contain large amount of non-coding and repetitive DNA. So coming to the organization of prokaryotes and eukaryotic genome, they slightly vary. Okay, you cannot make generalizations. Exceptions do happen, but generally speaking, prokaryotes have a single circular chromosome, and they are being condensed together into the cell by supercoiling by some proteins called the architectural proteins, and these prokaryotic DNA will interact with the cytoplasm, so transcription and translation happens together, and usually they are being considered, that is, the prokaryotes are considered to be haploid, and most of the non-essential genes have been coated in extrachromosomal plasmids. Okay, and they usually contain little of repetitive DNA in the case of prokaryotes. And I hope prokaryotic genome organization is clear to you. Of course, we found some variations of that also. Thank you.